Thank you for joining me on this webinar on segmentation building. Segmentations are very important operations in Video Media Lab software and in imaging in general. And I will go through how to build such segmentations in practice using the Video Media Lab software. I expect that you have some experience in transformation building, but if not, then you can also follow this webinar just as long as you follow the step-by-step approaches that I take. So first I would like to explain segmentations. They divide an image, as you can see here, the wheat kernels on a bluish background in the center of this slide, and make wheat pixels into white, which is the foreground, and the bluish pixels into black, which is the background. So the output is a binary image with only foreground or background in every pixel, and that shows us where the wheat kernels are and where we want to measure. Typically we would like to ignore the black pixels, the background, because that's not relevant for our sample. So what is interesting, where to measure, that's what segmentations will show, and they are frequently used as part of other operations that counts or measures the color or measures all kinds of other features on images. So segmentations are created in Video Media Lab software using the MSI menu, and you go down to the Segmentation Builder 2 menu point, and I will show you examples of exactly how to do this. First we will go to segmentation of wheat kernels in a petri dish and show you all the steps involved. Here we have a petri dish with wheat kernels, and we want to be able to segment out the wheat kernels from the background from the petri dish so that we can focus on just the wheat kernels. In order to do this, we first use a way to create contrast between the wheat kernels and the background, and we do that by our transformation builder. The preferred method is NCDA, which will take the wheat kernel, and then we will paint two layers that we want to differentiate. And in order to paint the layers, we click on the layer manager in the toolbar up here, and we use typically the first two layers. So I will paint the red layer as what I consider my foreground. I have a paintbrush here that I typically like. That's the paintbrush with 20 pixels, and if I want to choose a bigger one, I can choose this one or go back to 20 pixels. So I will typically paint not by brushing a lot of pixels here, but I will just spot the dark areas of the kernels, the white areas of the kernels, and then maybe some of the brownish and so on. So kind of being a representative for the expression of the wheat kernels rather than being exhaustive in the painting. I can zoom in and detect different areas here, and here is a very dark area that I could include. Okay, that was the foreground. I will also paint the background. I will be sure to paint something here in the very dark region of the background, and I will also paint out in another region where we have a light blue, and I will paint here also the small shadow that each kernel can have on the petri dish is painted. And of course we can also paint on the outside, because as you see here we have a stretch of a white line that we need to remove in order to not interfere with the kernels on the perimeter of the petri dish. There will be some ghost images, as you see here, in the wall of the petri dish, and we will remove them in another way. They will have the same color as the kernels, but will be not measured since we don't have a physical kernel there, but just a mirror image. And that goes all the way around the edge. So let's take this painting and try to press calculate here, and I will get an eigenvalue which is more than 100. So I have separated what I painted red from what I painted green with more than 100 standard deviations, which seems to be quite a good result. I could go in and try to apply that on my image. You'll see here that I get this kind of heat map for wheat versus background, and I can select the color bar to see the scale. You see that the yellow, red, orange pixels are positive, centered around +1, and the background is blue turquoise, centered around -1. And on the edge here, you see that the ghost pixels will get something higher value than the background, and that is something we need to deal with in the segmentation. So I have also a slight salt and pepper noise when I zoom in, and I would probably prefer maybe just smoothing a little bit in this case where I just want the segmentation. So I choose a Gaussian post-processing with a filter size of 5 pixels. So this is like 150 micron. And if I apply that to my image, then you will see a slight smoothing of the result, so that I have a more clear green line separating foreground and background. And you see the green line is the neutral color between foreground and background, and that's pretty good that we have a well-defined boundary here. So let's try that out. We save the transformation that enhances the contrast. I call it wheat versus background. Save it, and I remove the images here, and now I can go into Segmentation Builder 2, and this is kind of a pipeline for constructing a segmentation between the foreground and the background. So the first thing we want to do is our pre-segmentation. I press here, and you can select between a fixed mask and a petri dish segmentation. Since we do have the petri dish, we select the petri dish segmentation. We choose automatic, and we can press apply. So now we are segmenting out the petri dish, and you see here the interior of the petri dish as a new image, which is not so illustrative. I would rather choose another option down here. If we choose the arrow next to apply button, you see we can apply as a layer, and I could apply it as a red layer. So now you will see what the petri dish segmentation ended up with as the first segmentation, segmenting out the interior part of the petri dish. And if we look at that, then we now have a good starting point for going further and removing the background. And for this purpose, we take on the foreground background segmentation, and we need a transformation file to enhance the contrast between foreground and background. And that's good because that's what we just made. That was the wheat versus background. I select that, and we want to threshold that around 0, which is the good neutral value between foreground and background. I will apply this again first as an image. You can see the kernels here, but maybe more instructively again as the red layer. So now you see that wheat kernels are now marked with red, and we just have one final issue, that is that the wheat kernels are touching each other, so we don't have a good clear-cut separation between wheat kernels, and we would also like that as part of our segmentation. So we click on sequence step down here called separate touching objects, and we choose the adaptive parameters so we don't have to set them. The algorithm will automatically set it from the image or learn it from the image, and if we do that, I can again apply the segmentation as my red layer, and then we can actually go in and see that a line has been drawn between the touching kernels, so that we can now have a separation of kernels in order to know the size of each kernel, for instance, or shape of each kernel, and so on. So now we have a complete separation. We want to save this. And we call that segmentation weed kernels versus background. So just to note that we have also separated the individual kernels, and I save that segmentation, and now I'm actually done with my segmentation. But just in order to illustrate how it could be used, we can already apply the segmentation from up here called weed kernels versus background. So now I calculate a foreground mask showing what is interesting for me at this point. The background is still present. If I click on the mask, you can see that the background pixels are already or still there, but the foreground mask will tell you which pixels are actually interesting for me at this point. So we could make the red mask invisible so that we just see the weed kernels here. Now we could utilize the segmentation within the blob tool. Blob tool is a database of blobs or kernels, and we start with an empty database but can fill it up by extracting from this image. We extract from the image, and we don't have to actually put anything in here, because if we don't put any parameters, then it will use the foreground as the segmentation, and I just need to press apply, and that will import all the kernels into the blob tool. So you see all the seeds here, all the grains, and you see that we have 215 grains, and there are a few which are just dust particles, and if I want to change that, I see 2 dust particles. I can actually detect them by sort by area. So you see here that there are 2 small dust particles, or parts from the maybe the edge of the petri dish, and I can remove them here simply by marking them and right-clicking and pressing remove. Then they are gone. But I could also go back into my segmentation. So let's try that. I go back into the segmentation builder. I click load, and I load my segmentation from before. I can continue working on that, and I can select the last step, blob filtering, and filter all the blobs or kernels that I have by size. So I say I will take away anything lower than 1 square millimeter, and I can apply that, and that will mean that I do not get these very small kernels. I can save again on top of my segmentation, and then I will go back to the blob tool, and I will use another way. I can show you here. I can browse and choose my segmentation instead of just taking the foreground, and then I can extract all the blobs. And in this case I will get 2 less blobs because the 2 very small blobs have been taken out automatically, and I should get 213 blobs, as you see here. Click on any blob and get area, shape parameters, color parameters, and so on, and you can get a lot more, and that is the topic of the blob tool webinar. So now let's try to see if this segmentation can be used on barley kernels, which look quite different and slightly less regular than wheat kernels. Here we have an image of barley kernels in a petri dish, and the objective is to separate the kernels from the background and the petri dish so that we can quantify any property of the barley kernels only. Instead of making a new segmentation from scratch, we will try to see if we can use the pre-made segmentation made for wheat, and that we have in our MSI library. We choose the user-defined operations and the wheat kernel versus barley, and I can apply it here from the MSI library, and that will generate a new image with the segmented barley kernels, as you see here. But instead of using it like this, I could also go ahead and try out to apply it as a foreground. So I will remove everything but the barley kernels, and that will leave me with a nice foreground image here where you see that the barley kernels have been separated even though they touch. That means we can now do analysis of the barley kernels. So even though this segmentation was made for wheat, it actually works okay for barley, and we can see if we can import the barley into the blob tool. And in order to do that, we just start the blob tool, extract the barley kernels from the image, and we will probably get a number of smaller particles, which are the pieces of the barley kernels which are slightly irregular. So here we see all the barley kernels and some bits and pieces. If we now sort them by area, we can see that the bits and pieces are up here, and those can be removed, and then we have the barley kernels that is in the image, including some quite irregular ones. And now we can measure the barley kernels, and we can also see where a given barley kernel is located in the image. So now we move on to another type of application where we want to study pycnidia in wheat leaves. Here we see a wheat leaf with pycnidia on it. Pycnidia are asexual fruiting bodies from a fungus, and they are covering part of this leaf, and we want to be able to count them. In order to count them, we need to segment them out from the background of the leaf. The first thing we do is try to remove the petri dish and the blue background, and I have a fast button up here in the masking, so we simply mask the petri dish and blue background. Now we only have the leaf, and in order to do analysis on the leaf, then we try to mark the leaf with a layer. So I choose the red layer in the Layer Manager, I right click and I import the foreground pixels as the red layer, so all the foreground pixels are painted red. So that means I can now try to do analysis within the red. So the first thing I want to do is try to make a transformation that enhances the pygmedia. The pygmedia are not easy to paint because they are small, and in principle we could do it and then use the NCDA, but rather we would try to use a transformation that automatically tries to enhance the pygmedia. So for this purpose we use the MNF, and we do it on all pixels within the red layer, so the whole leaf. And we want to we do a small 5x5 MNF, so that means we see deviations within a 5x5 neighborhood as our noise here, and the signal is basically the whole variation across the leaf. And just to highlight how that should work out, let's try to calculate the MNF and apply it to our leaf. So here you see the first MNF, which is more or less like a monochrome image of our leaf. I can maybe enhance a little bit by use the foreground mask for autoscale, so it becomes a little bit more clear. I can also try to scale it with Six Sigma instead of min max scaling. That didn't help much, but maybe I should move on to one of the next MNFs, because typically MNFs 2, 3, 4, 5 could be the most important ones. Let's look at number 2. It does see some structure here within the infected area of the leaf, but not the pygmedia. And the next one has a slight imprint from the pygmedia, but it's not more clear than the first one. And then we move to number 4, and suddenly it seems like we get a good transformation for detecting pygmedia. So MNF number 4 should probably be our preferred choice. We can just have a look at number 5 and see if that does anything more for us, and it doesn't seem to be the case. So we run back to MNF number 4 and try to use that. So I will go to this graph here and right click, select none, and just select number 4. And we can see here the loading of number 4. So it has red and cyan colors minus blue and green yellow colors, and minus some near infrared as well. So quite an interesting loading vector here. Let's save it, not just number 4, and we call it pygmedia MNF 4, just to highlight how it was created, and I will save that in my MSI library. I will then go in and maybe try to have a little bit look about the dynamics around the pygmedia. So I choose the image tools, measurement, profile, and I draw a profile through a couple of pygmedia here, maybe 3. So you see them as bright spots here, pygmedia on a varying background, but you see the spots here, they have like minus 27, and outside the spots it's minus 32. So maybe a peak of 5 pixel values. So we are looking for peaks of maybe around 5, and maybe a little less too, because well, let's run through slightly more vague pygmedia, and we see that maybe here it's only peaks of 3 or 4. So let's try to look for peaks of 3 to 5 in this image, and this is done by making the segmentation now in Segmentation Builder. We take the image and we choose the foreground background segmentation. As transformation we choose the MNF4, and as band we choose the first, and we don't actually choose the threshold segmentation, but we now go to the second option here, H-dome segmentation, because we are looking for peaks, and we have quite a varying background. So we are interested in peaks, and we know that the height of the peaks is maybe 3 to 5. Let's try to put 4 in here, and let's see how it goes. I will just say here, we will choose the right image and apply, and you will see here that the pycnidia indeed are found. We should probably choose a threshold which is greater than 0, so we don't get too much response. Let me just try that. Now we see that the pycnidia are detected, but the longitudinal structures in the leaf here are also detected, because they are also ridges in this image. So we want actually to be able to detect the peaks without detecting the ridges. And for that purpose we have the gradient over here. So the gradient will remove or suppress the ridges and keep the peaks of the gray level function here. So maybe we can try with a value of 0.1 here, just to give some parameter, and I will see if that does anything for us. Yes, indeed, that does keep the pycnidia, but doesn't give us the ridges. So indeed, that was a very effective function for pycnidia detection. We can try another value than 0.1. If we now choose 0.2 and apply that, you see that we will maybe slightly remove more pycnidia, but not get any other desired effect. So definitely we should not choose 0.2. We could also try something lower than 0.1, 0.05, and apply that, and that seems also to be a viable choice. If we go even lower, 0.01, then we will not get the necessary sort of suppression of the ridges. So let's go back to 0.1 here and choose that as our segmentation, and we can decide whether we should put a limit on the size of the pycnidia. So we say, okay, we want to remove the smallest one, but in this case it actually seems like the pycnidia are indeed real and quite well segmented actually in this image. So I can save my segmentation here, pycnidia on weed leaves, and then we have a new function for our library. If we want to check out how it looks on our leaf, then we could actually import the open image, the pycnidia image, as our red layer, and then we see here the pycnidia that are detected marked as red, and we can tick them on and off through the visible icon over here. So we can see if we have counted all of them, and maybe a few ones are missed here, but that probably will mean that we should change the threshold slightly in order to get all of the pycnidia there. I think the good thing about this is that we do not get any false responses, it seems, but not sure whether this is a pycnidia out here, but it could definitely be. So that was pycnidia or segmentation on a leaf, and shows how to find objects on a varying background. So we move on to colony counting. So this is a quite frequent application where we have some dish or plate that we use to grow microbial colonies, and then we want to count the colonies. In this image we have a colony filter that is used in order to count colonies on the plate here, and we see a faint yellow color where we do have colonies. We also see a grid pattern which is used by the manual operator to count, but which is just part of the background when you do automated counting. So in order to count, we need to segment out the colonies and then count afterwards, and that means the first step would be to build a segmentation. Let's have a look at the segmentation builder again. The segmentation builder will need a petri dish segmentation, which we have here, and I think we can apply it as a red layer here, so we can get easily the outer edge. Maybe we want a larger margin here, so we can, instead of automatic, choose manual and say we want 4.2 millimeter as margin, and apply that as I will just remove the foreground here and just show how the 4.2 millimeter margin looks. So that looks like quite okay for this. Then we need to have a foreground-background segmentation of the colonies, and in order to do that we need to use the transformation builder to get the optimal contrast. So in order to get the optimal contrast, we make the red layer invisible, and we try to paint colonies on the plate here without any background, but let's see if we can get a higher contrast on the colonies by choosing the right wavelength. We see here if we choose a blue wavelength, then the colonies are more clear than they are in RGB. So let me choose the green layer to paint the colonies. I paint both where we have grid spots and where we don't. So in this case we will be sure that we see colonies both where we have the dots of the grid pattern behind and where we do not have that. And then we also need to paint the background here, and we see that we have the background out here and here and here and here. I just paint a little bit around the plate, and now we can go back to the transformation builder and say we want to separate green from blue. Let me just see how that goes. Separation of more than 40 standard deviations seems to be good. I apply it to the filter, and it seems like we do have a very nice separation of colonies and background, and we do see all the colonies here. And I will then save my colonies versus background transformation. Save that and leave the transformation builder and come back to the segmentation builder. Here I'll just remove this and go back to view mode is RGB, so we see it as a color image. Now we can add the foreground background segmentation with our user-defined colonies versus background transformation. Threshold is 0, foreground is greater than 0, and we can now apply that either to a new image, as you see here, or we can apply it as the foreground of the image. So here you have the colonies marked, and there are several ways we can mark or count the colonies when we have them like this. One way would be to go into the blob tool and extract the blobs from the image with the foreground here. Here we see all the blobs. We have 42 colonies. We sort by area, and then you see all the colonies from the smallest to the largest colonies. 42 colonies altogether, and if we are happy with this result, then we can save it and call it colonies versus background, and now it's a segmentation. And this concludes our Segmentation Builder webinar, and we are ready to take your questions.